Welcome to Ultimate Sleep Doctor. Tonight, we'll journey to a world of incredible resilience. Let's consider the tardigrade, often called the water bear. These tiny creatures, barely visible to the naked eye, possess a secret: the ability to survive nearly anything. They've endured the vacuum of space, extreme temperatures, and intense radiation. Isn't it remarkable to think about such delicate beings holding such profound strength? Perhaps a reminder that even the smallest things can possess incredible power, and that survival often lies in quiet adaptation. If you find these explorations soothing, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. Just breathe. Let your thoughts drift like clouds across a still sky. Let's begin. The story of the tardigrade, or water bear, isn't just a tale of incredible resilience. It's a story stretching back nearly 500 million years. Their evolutionary history is a surprisingly complex puzzle, pieced together from the faintest of fossil clues and sophisticated scientific analyses. It began long before we even knew they existed, with the discovery of Ozark-Ozarachn in 2014. This remarkable find, unearthed in Arkansas, wasn't a typical tardigrade. It was an arthropod, a distant relative, possessing many tardigrade-like features, suggesting a lineage extending back to the Silurian period, approximately 435 million years ago. This immediately shifted our perspective, indicating that the group wasn't a relatively recent invention of evolution, but rather a branch that had been quietly persisting through immense geological change. The challenge, of course, is that tardigrades are tiny, usually less than a millimeter long, and their bodies are incredibly delicate. Fossilization is a rare event, and the processes of burial, compression, and alteration over millions of years have left their mark. Many purported tardigrade fossils are actually arthropods with similar features, highlighting the difficulty in definitively identifying ancient tardigrade remains. Scientists utilize a combination of morphological analysis, examining the physical structure of the fossils, alongside molecular data, comparing DNA sequences to build their phylogenetic trees. Early molecular studies leaned towards placing tardigrades closely related to arthropods, particularly the extinct group of marine arthropods known as marine harvestmen. However, as more data accumulated, particularly from more complete tardigrade genomes, the picture became more nuanced. The placement of tardigrades within the animal kingdom has been a source of ongoing debate. Some studies suggest a closer connection to nematodes, roundworms, representing a basal animal group. This hypothesis is supported by certain morphological characteristics and recent genomic analyses. Other research continues to point towards a more ancient position, potentially sharing a common ancestor with other groups like sponges or ctenophores, even more primitive animal phyla. Stable isotope analysis, examining the ratios of different elements within their tissues, provides a valuable tool for understanding their ecological roles. These studies suggest that tardigrades have occupied a remarkably diverse range of environments throughout their history, from marine sediments to freshwater springs, indicating a highly adaptable lifestyle. Major extinction events, such as the Permian-Triassic extinction, approximately 252 million years ago, undoubtedly impacted tardigrade populations. The sheer devastation likely reduced their numbers and shifted their distribution. However, tardigrades demonstrate an extraordinary ability to survive even catastrophic events, a key factor in their persistence. Their ton state, a dormant, dehydrated state, allows them to withstand extreme conditions, including radiation, desiccation, and even the vacuum of space, providing a buffer against these calamities. The ongoing search for even older tardigrade fossils is crucial. Discoveries of well-preserved specimens from earlier geological periods would provide invaluable insights into the origins of this resilient group, potentially pushing back their appearance by hundreds of millions of years, fundamentally altering our understanding of their evolutionary trajectory. The story of the tardigrade is far from complete, and each new fossil, each new data point, adds another fascinating chapter to this ancient tale. The remarkable survival strategies of tardigrades, often called water bears because of their sturdiness, are built around a phenomenal ability called cryptobiosis. It's like they're holding a secret, a way to essentially freeze or dehydrate themselves and wait for better times. This isn't just a simple sleep. It's a deeply complex, reversible state of suspended animation. Cryptobiosis actually encompasses several distinct forms, each tailored to specific environmental stresses. Anhydrobiosis, perhaps the most famous, is the ability to survive extreme desiccation or drying out. Tardigrades achieve this by drastically reducing their water content, a feat made possible by a fascinating combination of chemicals. They accumulate molecules like trehalose, a naturally occurring sugar, and glycerol, which act as compatible solutes, preventing the damage that would normally occur when water disappears. These solutes essentially shield their cellular structures, preserving their delicate proteins and DNA. Beyond simply reducing water, tardigrades modify their membranes, becoming more fluid and less prone to disruption. DNA repair pathways are also dramatically upregulated, constantly scavenging for and fixing any damage that might arise during this period of stress. The cellular metabolic rate plummets, conserving energy, a crucial factor in the survival of these tiny creatures. Cryobiosis, or cold cryptobiosis, is another form where tardigrades withstand freezing temperatures. At these extreme lows, their metabolism slows to a crawl, and they can remain viable for years. Research is ongoing to understand precisely how they do this, but it's clear that maintaining membrane integrity and preventing ice crystal formation are key. Osmobiosis represents the ability to survive extreme osmotic pressures, conditions of high salinity or water potential. Anaxibiosis allows them to endure oxygen deprivation, a strategy most evident in deep-sea tardigrades where oxygen levels are incredibly low. It's important to note that transitioning into cryptobiosis isn't a single event, but rather a precisely orchestrated cascade of biochemical reactions. Triggering the ton state, the hardened, protective shell they form, requires a significant energy investment. Cellular metabolic adjustments, including shifting fuel sources and activating specific stress response genes, are fundamental to this process. Interestingly, the formation of the ton is accompanied by a dramatic change in cuticle permeability. The cuticle, a protective outer layer, becomes virtually impermeable to water, further reinforcing the organism's ability to withstand dehydration. Researchers are continually investigating the precise molecular mechanisms involved in this transformation and the role of specific proteins in modulating permeability. The reversibility of cryptobiosis is equally remarkable. When conditions become favorable again, when water returns or temperatures rise, tardigrades meticulously rehydrate, restoring their normal metabolic function. This process isn't simply about adding water. It's about reactivating the complex network of enzymes and proteins that govern their life. Recent studies have shown that specific signals, perhaps related to changes in light or temperature, play a critical role in initiating the reverse transition. The potential applications of understanding and manipulating cryptobiosis are vast, ranging from preserving biological materials like organs and tissues to even protecting astronauts during long-duration space missions. Tardigrades, those tiny water bears, aren't just known for their incredible ability to survive extreme conditions, they're also surprisingly sophisticated eaters. Their feeding strategies are incredibly diverse, reflecting their varied lifestyles and the bounty of the environments they inhabit. We've discovered several distinct feeding types, largely determined by their body shapes. Some, like the raptorial tardigrades, have evolved specialized appendages, often called stylets, tipped with sharp, needle-like structures. These stylets are used to pierce the bodies of their prey, which can range from single-celled bacteria to tiny nematodes or even other, smaller tardigrades. The way they capture prey is astonishing. Some will actively pursue their meals with remarkable speed, while others employ an ambush strategy, patiently waiting for a passing organism. The morphology of these appendages is remarkable. Certain species, like Haplophthalmus species, utilize a grasping claw-like structure alongside the stylet, allowing them to firmly hold onto their slippery meals. Then there are the direct-feeding tardigrades, which simply engulf their prey, often utilizing their buccal arms, fleshy, tube-like structures, to draw in microorganisms. Their feeding appendages aren't just for capturing. They're equipped with specialized muscles that allow for precise movements and a secure grip, essential for tackling prey that can be significantly larger than themselves. But the story doesn't end with the external feeding mechanisms. Tardigrades also harbor a fascinating and largely unexplored world within their guts, a complex microbiome. For decades, scientists overlooked this critical element, but recent research has revealed that tardigrades maintain a symbiotic relationship with a diverse community of bacteria and archaea. These tiny guests play a crucial role in several key functions. The microbiome aids in the breakdown of complex carbohydrates and other organic matter, essentially helping the tardigrade digest its food more efficiently. Beyond digestion, the gut microbiome contributes significantly to the tardigrade's immune defense. Certain bacterial strains have been observed to directly inhibit the growth of pathogens, acting as a first line of defense against infection. Furthermore, the microbiome appears to be intricately linked to the tardigrade's ability to detoxify harmful compounds, assisting in the removal of toxins ingested through their diet. Scientists have begun to analyze the composition of these microbiomes, often using DNA sequencing to identify the specific bacterial species involved. They've discovered that the makeup of the microbiome isn't static. Tardigrades can actively manipulate their gut communities, shifting the balance of bacterial populations based on environmental cues, such as food availability or stress levels. For example, under nutrient-poor conditions, they seem to favor bacteria that are particularly efficient at extracting energy from scarce resources. Interestingly, the study of tardigrade microbe interactions has also illuminated the darker side of this relationship: host-parasite interactions. Certain bacteria can sometimes be detrimental, causing infections or disrupting the tardigrade's digestive processes. The ability of tardigrades to respond to these imbalances and actively manage their microbiome represents a sophisticated adaptation, contributing directly to their remarkable survival capabilities. Ongoing research promises to reveal even more about this hidden world within the water bear and how it truly underpins their resilience. Tardigrades, these tiny bear-like creatures, aren't just masters of survival in extreme conditions. They're also surprisingly complex reproducers. Their reproductive strategies are incredibly varied, reflecting their adaptability across diverse environments. Many species, particularly those in aquatic habitats like mosses and lichens, reproduce asexually, primarily through budding. A small outgrowth develops on the parent's body, eventually detaching to become a fully formed, miniature tardigrade. This process is remarkably quick, allowing rapid population growth when conditions are favorable. However, some tardigrades employ sexual reproduction, a more involved process where genetic material from two individuals combines. This typically occurs through external fertilization, where the female releases eggs and the male deposits sperm. The species Euborelia viridis, found in the deep sea, is a stunning example of this, with males actively seeking out females to deliver their sperm. Internal fertilization has also been observed in certain terrestrial species, offering enhanced protection for the developing embryo. The precise mechanisms governing this switch between asexual and sexual reproduction are still being investigated, but it's believed to be strongly influenced by environmental cues, particularly nutrient availability and temperature. A particularly fascinating aspect of tardigrade reproduction is their hermaphroditic nature. Most tardigrades possess both male and female reproductive organs, allowing them to self-fertilize under certain circumstances. This isn't always the case, though some species exhibit complex mating rituals involving tactile interactions and chemical signaling to facilitate successful cross-fertilization. The developmental pathways of tardigrades are equally diverse, heavily impacted by whether reproduction is sexual or asexual. Asexual development follows a simple pattern. Cell division, primarily through mitosis, creates identical copies of the parent. Sexual development, in contrast, involves a significantly more intricate sequence of events, beginning with the fusion of sperm and egg, initiating embryonic development, and ultimately leading to the formation of all the tissues and organs of the new tardigrade. Larval stages represent a critical transition in many species. Aquatic larvae, often resembling miniature versions of their parents, are typically free-swimming and possess specialized structures for feeding and movement. These larvae eventually undergo a metamorphic process, transforming into the adult form. Terrestrial larvae, found in environments like mosses, exhibit different adaptations, often including enhanced cuticle protection and feeding strategies suited to their terrestrial habitat. The genetic control of this development is fascinating. Research has identified key signaling pathways like WNT and BMP that are crucial for coordinating cell division, differentiation, and morphogenesis. Transcription factors, proteins that regulate gene expression, play a central role in determining the fate of different cells. Furthermore, recent studies have uncovered evidence of induced parthenogenesis, the ability of a female tardigrade to develop without fertilization. This process, observed in several species, involves activating dormant genes that would normally be expressed only in the presence of sperm, allowing for rapid reproduction when mates are scarce. The genomic implications of this process are still being unraveled, but it highlights the incredible plasticity of the tardigrade genome. Finally, environmental stressors can dramatically impact tardigrade reproduction and development. Exposure to heat, radiation, or desiccation can trigger changes in gene expression, influencing developmental timing and potentially promoting asexual reproduction as a survival strategy. The ability of these tiny creatures to adapt and overcome such challenges continues to astonish scientists and underscores the remarkable resilience of the tardigrade lineage. The world of a tardigrade is a quiet one, a carefully orchestrated dance of survival dictated by a surprisingly sophisticated suite of behaviors. These tiny creatures, often only a millimeter long, aren't simply waiting for a favorable condition to arise. They actively navigate their surroundings, reacting to a constant stream of sensory information. Their movement is remarkable, considering their size. They can walk with a jerky, yet purposeful, gait across mossy surfaces, propelled by their 8 stubby legs. Swimming is achieved through rhythmic undulations of their bodies, a surprisingly agile movement in aquatic environments. Crawling is a common strategy, utilizing their adhesive feet to grip onto almost any surface, from damp leaves to the scales of a reptile. The neurological control of these movements is still being investigated, but research suggests a decentralized nervous system, with neurons distributed throughout their bodies, allowing for rapid responses to stimuli. A key aspect of their behavior is their sensitivity to touch. Tardigrades possess a network of mechanoreceptors, tiny sensory structures, distributed across their bodies. These receptors, triggered by physical contact, transmit information about their environment, allowing them to avoid obstacles and even detect the presence of potential predators. They can sense vibrations in the substrate, providing early warning of approaching danger, and respond with a burst of movement to escape. Furthermore, the precise arrangement of these sensory neurons isn't static. It's adaptable, changing in response to experience, suggesting a form of learning. Perhaps one of the most extraordinary sensory organs in the tardigrade world is the Johnston's organ. Located on their first few legs, this complex structure is exquisitely sensitive to changes in gravity. It acts like a miniature seismograph, detecting subtle shifts in the gravitational field. This ability is crucial for navigation, helping them maintain their orientation, and even for finding stable surfaces to cling to. Scientists believe the organ uses a fluid-filled chamber and a network of hair-like structures to transduce gravitational forces into nerve signals. Beyond touch and gravity, tardigrades respond to light, although their vision is rudimentary. They seem to be particularly sensitive to blue light, potentially using it for orientation, or perhaps even for signaling. Chemical cues also play a vital role. They can detect gradients of nutrients, guiding them towards food sources, and they can detect volatile organic compounds released by plants or other organisms, communicating information about their surroundings. Interestingly, tardigrades exhibit rhythmic behaviors, suggesting the existence of internal biological clocks. Research has revealed that they display cyclical changes in activity levels, particularly during periods of darkness, and these cycles seem to be synchronized with the 24-hour cycle of light and dark. The precise mechanisms regulating these circadian rhythms are still under investigation, but they likely play a role in optimizing their energy expenditure and maximizing their chances of survival. Understanding these internal rhythms is a key step in unlocking the secrets of their incredible resilience. Their complex behaviors, driven by a finely tuned sensory apparatus, represent a miniature marvel of adaptation and survival. The world of tardigrades, those incredible water bears, isn't confined to any single habitat. They've become remarkably widespread, a testament to their extraordinary ability to survive in some of the most challenging conditions imaginable. Scientists estimate their global distribution is astonishingly broad, with specimens discovered in nearly every corner of the planet. You'll find them thriving in the frigid, icy depths of the Arctic and Antarctic, clinging to mosses and lichens in the high alpine regions of the Himalayas, and even nestled within the sandy dunes of the Sahara Desert. Their adaptability is truly remarkable. Surprisingly, tardigrades have also ventured into environments considered utterly hostile to most life. Deep-sea hydrothermal vents, where superheated, mineral-rich water spews from the Earth's crust, support communities of these resilient creatures. They've been found amongst the tube worms and bizarre invertebrates that call these vents home, tolerating extreme temperatures and pressures. Even further afield, researchers have detected tardigrades on the International Space Station, demonstrating their capacity to survive the stresses of space travel. This suggests a potential for long-term survival in conditions that would be immediately lethal to most organisms. However, their survival isn't just about enduring extremes. It's about finding the right balance of resources. Temperature, humidity, radiation levels, and the availability of nutrients all play a critical role in determining where they can flourish. Generally, they prefer environments with moderate moisture and a steady supply of organic matter, often found in soils and mosses. Yet they demonstrate a surprising tolerance to dehydration, entering a cryptobiotic state, a dormant, essentially lifeless state, when exposed to harsh conditions, allowing them to conserve water and protect their vital tissues. The tardigrade's remarkable resilience is increasingly recognized as a valuable tool in environmental monitoring. They are being used as bioindicators, meaning their presence or absence, as well as their physiological state, can provide insights into the health of an ecosystem. Because they're sensitive to pollutants and environmental stressors, a decline in tardigrade populations can signal problems with water quality, soil contamination, or overall ecosystem disturbance. Researchers are carefully analyzing tardigrade populations in polluted areas to assess the impact of human activity. Their ability to withstand high levels of radiation, far exceeding what's harmful to humans, is another fascinating aspect of their biology. Studies have shown that tardigrades can repair DNA damage caused by radiation, a crucial adaptation for survival in areas exposed to nuclear fallout or other radiation sources. Furthermore, their unique physiology allows them to survive prolonged periods of desiccation, radiation exposure, and even vacuum conditions, all factors that contribute to their remarkable adaptability. Despite their resilience, human activities are beginning to impact tardigrade populations. Pollution from agricultural runoff, industrial discharge, and plastic contamination pose a significant threat. Climate change, with its associated shifts in temperature and precipitation patterns, is also altering their habitats and potentially disrupting their survival. Ongoing research into global biodiversity data, utilizing DNA sequencing and distribution mapping, is helping scientists understand the full extent of the threats and develop strategies for conservation. It's a reminder that even the most extraordinary creatures can be vulnerable in a changing world, and that understanding their secrets is vital to protecting the delicate balance of our planet. Tardigrades, those tiny, resilient creatures, aren't just found in moss and lichen. They've also ventured into some of the most hostile environments imaginable. Their survival secrets are truly astonishing, particularly when it comes to enduring the effects of radiation and the challenges of space. Scientists are still unraveling the precise mechanisms, but we're beginning to understand how these microscopic bears can shrug off doses of radiation that would obliterate most life forms. A key component of their defense is a sugar called trehalose. When a tardigrade enters a state called cryptobiosis, a kind of deep sleep, trehalose replaces water in their cells. This creates a stable, glassy matrix that protects their proteins and DNA from damage, much like antifreeze protects delicate tissues in a freezing environment. During radiation exposure, this trehalose shield works incredibly effectively, minimizing the impact of the energetic particles. But trehalose isn't the whole story. Tardigrades also possess remarkably efficient DNA repair pathways. They can quickly identify and fix damaged DNA strands, essentially patching up the damage before it can cause catastrophic problems. Researchers have observed that tardigrade cells have a significantly higher capacity for DNA repair compared to many other organisms. This ability complements the trehalose protection, providing a double layer of defense. Another fascinating process is called nucleoid condensation. The DNA within a tardigrade cell isn't just floating around. It's tightly wound into a compact, spherical structure, the nucleoid, and this condensation shields it from radiation and physical stress. It's like rolling up a sweater to protect it from getting snagged. The tighter the wrapping, the less vulnerable the DNA becomes. Now, let's consider space. The International Space Station has become a unique laboratory for studying tardigrades under extreme conditions. Experiments have demonstrated that tardigrades can survive exposure to the vacuum of space, extreme temperature fluctuations, and, crucially, high levels of radiation, far more intense than they would typically encounter on Earth. They enter cryptobiosis during these exposures, further enhancing their resilience. Data collected from these experiments shows that tardigrades can remain viable for extended periods in space, exhibiting signs of metabolic activity and even attempting to reproduce after returning to Earth. This suggests a level of adaptability previously unimaginable for organisms of their size. The rapid entry and exit from cryptobiosis, facilitated by space conditions, appears to be a critical factor in their survival. The implications of these findings extend beyond simply understanding a remarkable animal. The ability of tardigrades to withstand such harsh conditions raises the intriguing possibility of life existing in other extreme environments, perhaps even on other planets. If life can survive the radiation and vacuum of space, then the chances of finding life on worlds like Europa or Enceladus, with their icy surfaces and potential subsurface oceans, suddenly seem much more plausible. Further research into tardigrade adaptations could provide valuable insights into the limits of life and guide the search for extraterrestrial organisms. They are, in essence, tiny ambassadors of resilience, offering a glimpse into the potential for life to thrive in the most unexpected corners of the universe. The scientific community is currently buzzing with activity around tardigrades, those incredibly resilient microscopic animals. Research efforts are exploding, driven by a deep fascination with their extraordinary ability to withstand conditions that would be instantly fatal to almost any other life form. A core component of this research involves detailed genomic and proteomic analyses, allowing scientists to decipher the precise mechanisms behind their survival. They're essentially building a comprehensive instruction manual for resilience, examining every gene and protein involved in repairing damage and entering a state of suspended animation, or cryptobiosis. One particularly exciting area of investigation revolves around the enzymes produced by tardigrades. These enzymes, especially those involved in DNA repair, are showing remarkable potential for industrial applications. Imagine using tardigrade-derived enzymes to create more efficient and sustainable manufacturing processes, or even developing new cleaning agents that are both powerful and environmentally friendly. Researchers are actively exploring how these enzymes can be adapted for use in various sectors, driven by their superior stability and effectiveness under extreme conditions. Beyond industrial applications, tardigrade DNA itself is generating considerable interest, primarily within the field of synthetic biology. The DNA is remarkably stable and contains unique genetic sequences that could be harnessed to create entirely new biological systems. Scientists are investigating the possibility of using tardigrade genes to build novel enzymes or even create entirely new forms of life, a concept that's both thrilling and raises profound ethical questions. Furthermore, research is focusing on understanding the intricate biochemical pathways that allow tardigrades to enter and exit cryptobiosis. This state of suspended animation, triggered by dehydration, radiation, or other stressors, involves a remarkable reduction in metabolic activity and the formation of protective compounds like trehalose, which stabilizes cell membranes. Understanding this process could have significant implications for preserving organs for transplantation, extending the shelf life of food, or even developing new strategies for human hibernation. The study of tardigrade physiology isn't limited to simply identifying survival mechanisms. It's also shedding light on the broader evolutionary history of life on Earth. Tardigrades are incredibly ancient. Traces of similar organisms have been found in the fossil record dating back over 500 million years. Their unique adaptations suggest a deep evolutionary history, providing invaluable insights into the early diversification of life and the strategies that organisms have employed to cope with environmental challenges. Looking ahead, researchers are planning increasingly sophisticated experiments, including long-duration space missions with tardigrades to further investigate their physiological responses to the extreme conditions of space. Analyzing these responses, combined with detailed genomic data, will undoubtedly reveal even more secrets about their survival strategies. Finally, the study of tardigrades serves as a powerful reminder of the resilience inherent in life itself, a concept that becomes increasingly relevant as we face the challenges of a changing planet and consider how we can best preserve biological diversity for generations to come. If you're still awake and enjoyed this exploration, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. The tiny resilience of the water bears has now slipped away, their incredible story resting gently within you. Let your thoughts drift, calm and unhurried, like the slow currents of a quiet stream until sleep finds you. Good night.